Good afternoon, everybody. We are really happy to start this series of scientific webinars here at St. Peter's School. Today's webinar is about biotechnology and genome editing. And our keynote speaker is Dr. Pareras.
And what a wonderful opportunity to talk about science as well. To talk about the science means to talk about the truth. In this era of the fake news, etc., I think we all have a responsibility to be more science-focused and more data-driven. So congratulations for this webinar. Literally, you're going to live amazing things in your life. This is only starting what I will present today. A revolution is coming, and I really envy you. So the most important thing that I want you to understand in this lecture is that life is programmable. Okay, this is the assumption behind all gene therapy, gene editing, and synthetic biology. And today's class is going to be about synthetic biology, precisely, CRISPR and synthetic biology. Okay, so what happened in the last 70 years in medicine that has been so extraordinary? Just 70 years ago, in 1953, Watson and Crick discovered the DNA structure. They first described the way that life passes on the information to the next generation. Just a little bit after them discovering this DNA structure, the medical community started finding out that a lot of diseases were genome related, were diseases because there were mistakes on the DNA. And then it opened the field to three successive fields that are really redefining what medicine can do. And this is truly a revolution, okay? So first we had gene therapy. And gene therapy is just the transfer of genetic material to a patient. Let's imagine, for example, the whole genome, your genome, okay? It has roughly about 35,000 genes. Imagine like a library with 35,000 books, okay? So gene therapy would be the equivalent of taking out one book, every book is a gene, okay? One book that is badly written, that has some problem in it, and then replacing it with another book, okay? This is exactly what gene therapy does. In fact, what it does is we don't take away the book, we just add a new one that has the correct instructions for the protein that this gene produces, and then the protein is produced inside the body, and then everybody's happy because things start to work again. But this is very cumbersome because, as you can imagine, a gene is long, and then you need to change the whole book. And then someone invented, and we're going to talk about that a little bit later, someone invented a way of edit the genome, not just changing one gene with another, but getting inside the gene and change the letters that are wrong. It would be the equivalent of a word editor. It's like using Microsoft Word, but for a gene, okay? So now with the library analogy, we're not just taking out one book and replacing it with another. We're just opening the book. We're finding the sentence that is wrong because it's usually a very short sentence. Sometimes 30% of the time it's just one letter that is wrong, okay? Sometimes it's a sentence, okay? But we go inside the gene, in this case the book, and we change the sentence, okay? So the mutation is corrected in situ, in the place that is wrong. But we weren't happy with that. And then we are starting now what's going to be the most important revolution of the 21st century, which is going to be synthetic biology. It is not just that we correct now a gene or edit the sentence. Now we are programming a new book. We're introducing a new book inside the library just to make the book do things that were unthinkable before, okay? So now we are programming life. We are, in a way as well, creating life, because as you will see in a few slides, we can create very basic forms of life already, and that's indeed the future of medicine. For example, we can program cells to do something. In this case, we could program cells to produce something, okay? So we will have some cells, we will introduce the gene, the new gene that codifies, for example, for the production of fuel or of light or of new garments, the cell as a living factory, okay? So large-scale factories. This is being done. This is not science fiction. This is already being done. We can create, for example, artificial silk. We can create garments, for example, with cells that change their color depending on the light, just as chameleons do in real life, because the gene is exactly the same one. Imagine, for example, a coat in the future that could change color. This is being done as we speak today. We have, for example, cells that we program them to emit light. We could also use the cells to introduce them inside humans to solve some disease. Imagine, for example, this is being done very extensively. For example, in healthcare, we could program T cells to fight tumors, for example, and this is being extensively done through CAR-T technology. Maybe you have heard about it, chimeric antigen receptors technology. We can program cells to fight against infections. We could program cells in the field of immunology. We can program cells to detect toxins, for example, inside the body and fight against those toxins, etc., etc. Okay. Then the third field that we are starting as well in synthetic biology is to release on the environment these cells to do something. For example, in bioremediation, this is very useful. Imagine a cell that is programmed to eat all the plastic in the ocean. The same with pollution, the same with biomining, a very interesting field, just to extract metals in a much more productive way. Food production, you have all heard this artificial meat revolution that's coming on. This is done precisely with synthetic biology tools and technologies. We could even use DNA in this case to store, as if it were a hard drive, okay, to store information. Of course, it has a problem that to access the data and to record the data is not very fast at the moment, but a lot of people are working in the world very hard to make it really, because the great advantage is that it would last a lot of time, and every DNA molecule can hold a lot of information. This is the paper that was published in 1953 by Watson and Crick, the molecular structure of nucleic acids. I'm just introducing it in the lecture because I want you to really go to the source always. When you hear something related to science, don't just stay with what the speaker, in this case myself, says. You should go to the source and check and read it, okay? So this is the original paper. Could you project the next slide? And this is exactly how CRISPR works. The CRISPR is a construct made by two parts. It's the single guide RNA, which is the part of the CRISPR system that locates the place where we want to do the edit, okay? And then the Cas9 nucleases, which is a protein that is designed as a scissor. I mean, it makes the cut exactly where the guide RNA tells the Cas9 to do the cut. Okay, there are many types of Cas. This is a more complex slide, and we won't get into biological details, but many different types of Cas. The Cas9 is a more usual one, but then you have the CPF1s, also known as Cas12. You have dead Cas, which means that you can interfere with the gene without changing it. We have Cas9 nicases, which just produce one, I mean, not a double strand break, but one strand break. There are many variants to do that. But in essence, what happens is what you can see at the right of the slide, the place of the cut is detected by the guide RNA and one thing that's called the PAM that's in the original DNA. Then the guide RNA binds to the DNA, then the Cas9 undergoes a conformational change and becomes the scissor that we want, and then it produces the double strand break. This DSB, double strand break, is essential because from that moment on, the cell tries to repair, because the cell is programmed by millions of years of nature evolution to repair that cell. Then you could have, for example, the non-homologous end joining, which would mean that the both extremes of the cut are repaired immediately. But if you provide a template of the gene that you want to edit, sometimes you get this homology-directed repair, which is the other mechanism the genome has to repair, that introduces this strand of DNA in the place where it's needed just to make the correction. This means that in any gene therapy, or in any gene editing in this case, you will have always cells that have underwent the correction and cells that haven't, but sometimes with just as few percentage of cells as 5% of the cells corrected, that means that the disease disappears. This guide RNA has two parts: the red one, which is called the spacer, or CRISPR RNA, CRRNA, and then the scaffold, which binds to the Cas protein. Okay, this spacer is just 20 nucleotides long, and just with these 20 nucleotides, we are able to find exactly the place where we want to do this cut. Of course, if these 20 nucleotides are repeated elsewhere in the genome, we could have a CRISPR cut in some other place that's not our intended place to cut, and this is called an off-target effect. And this is the major problem that CRISPR-Cas systems have right now, but we are trying really hard to solve that problem just to make it even more precise. It is already very precise, don't misunderstand me, but we can make it even more precise. I give you here the picture of the first living thing that escaped the evolutionary tree, but Darwin is dead. There's no longer, once this happened, Darwinism is dead because we are controlling the evolution. This is a picture of something that Craig Venter did just 10 years ago, which is the creation of a cell from scratch. This cell was constructed with programmed genes, with a minimum set of genes that make a cell viable. Only 400 genes make a cell viable. If you take away one gene of this cell doesn't live. So we got it precisely for the right amount of genes that make that thing not only live, but grow, eat, and divide. Okay, so this is an artificial cell that was constructed just programming with the 4 letters of the DNA code. We are not even limited by this. I want you to understand that until very recently, the boundaries of biology were set by nature, but now these boundaries are set by scientists and human beings, because this is the first paper published with a synthetic DNA that was using not just the 4 original letters, but an extra 2 letters. This is what I like to call the first alien being on Earth, because this is not the result of evolution. This was precisely created with new programming code. Okay, this paper is amazing. I really recommend you to read it. And this is my final slide. And I want for you to think right now what we should do. And I'm going to ask you, all of you, through the chat to let me know what's your opinion. But listen, in this 2x2 matrix, I try to summarize what we can do today with genome editing. Okay, and this is a moral dilemma. Okay, this is an ethical issue. We could use genome editing for therapy things, meaning to correct diseases, or we could use editing to enhance the human being. And at the same time, we could do editing just on somatic cells, meaning that this editing that we do doesn't get to our descendants, to our sons and daughters, and to their sons and daughters. Or we could do editing in the germline, exactly as what happened, for example, in China with this crazy scientist that already has done CRISPR intervention in 2 kids that were born and are alive. A is the option where you are only comfortable with correcting diseases and in somatic cells. That means the green area that you have here in the slide. Okay, that would be A. B would be doing therapy not only in somatic cells, but in the germline as well. Meaning that if we're going to correct a rare disease, let's do it forever, not just for this generation, but let's remove the genes for all their descendants, okay? Option C is doing editing as well for enhancement, but only in somatic cells. That means, for example, we could improve humans now, better individuals. And then option D would be the full spectrum, would be doing enhancement as well on the germlines, which means precisely that Darwin is dead, rewriting evolution, okay? And this, of course, is not being done now, but would you be comfortable in the human race doing that, controlling its own destiny by rewriting evolution's rules? So I would love you to vote, each one of you, which of the letters would you feel comfortable with? I would love someone from the teachers to tell me what the results are. Not the exact results, but more or less which is the option that is winning.
So Joan, Mauri, maybe you can monitor the chat. Joan?
At the moment, B was winning.
Let me tell you my opinion now, but this is everything that I explained until now. It's science, okay? Now I'm going to express an opinion, which is not the truth. It's just my personal opinion, okay? Because there are debates, there are very long debates about this issue in medicine right now, okay? My opinion, my way of thinking is always safety first. Safety first means we can only perform the changes where we know exactly what we're doing. We can't experiment at this point doing very crazy things. So at this point, I'm comfortable with A, okay? And I would even be comfortable in a few years, once we know a little bit more, with B as well. Because I think a rare disease, if it's of genetic origin, we should be able to change it once and forever, you know? So there's nothing wrong with correcting a disease forever. The problem is, are we causing any harm in future kids precisely by doing that? And as we don't know, I prefer to be prudent. But let me tell you something. In China, they're doing B and C like crazy. I mean, in fact, CRISPR is so easy to perform that it could be done in a garage, literally. So this is coming. For example, anyone thinking we should enhance, well, why not? Plastic surgeons have been enhancing for more than 50 years, or ophthalmologists have been enhancing. And what we accept as society depends on what we consider as normal. You know, what is normal? Maybe normality, the concept of normality will shift in this century, you know, because what was normal maybe 20 years ago, what wasn't normal 20 years ago, maybe today it is normal. And then maybe 20 years from now, it will be normal. So let's not assume things. This is an ongoing debate. Society is going to debate it quite hard in the coming years. But what's for sure is that this is a revolution. This never happened in human history. The ability to program life, to make life a product. And this is going to change things. I bet you that the Amazons, Teslas, and Apples of the future, of 10 years' time, are going to be synthetic biology companies. And I'm ready to take any question. I'll be more than happy to answer if I know the answer, because first and foremost, what I am is a scientist, okay? And scientists, we only deal with the truth or what the things that we know. If we don't know them, we say we don't know them, okay?
Mi nombre no. Yo tenía curiosidad por saber si en este ambiente científico de desarrollos y todo esto es un ambiente muy competitivo o predomina más la colaboración entre científicos. Gracias.
Wow, what a great question, Ruth. This is a very important question, very interesting question. Listen, it's very difficult to answer because among scientists, when you consider only science, there's a lot of cooperation, but there's a lot of competition as well, because all good scientists want to publish first in Nature or in Science or in the Lancet or the New England Journal of Medicine their findings. Okay, so it is ultra competitive, but it is, I would say, a very healthy competition, because they compete, but scientists are very trained to admit and to quote everything that they learn from elsewhere. And I would say it's a very, very intense competition, but a very healthy one as well, because they are very fair. It's not like companies competing, you know. What I see when I go out is people competing really hard, but in a very fair way, just pretending to be the best at what they do. And I think that's great. That's what moved forward science. Yes.
I would like to ask you if you know, or if you think that there will be a vaccine for COVID-19, and when?
Super interesting. Could you project my last slide, Teresa? Listen, this is, again, a very important question, and I can give you full certainty on my answer, because the correct answer is I don't know. Okay, there are viruses that we have tried long time to get a vaccine. For example, HIV, the AIDS virus, and we never succeeded. So there are viruses which is very difficult, or I would say even impossible, to get a vaccine. Other viruses are very easy to tame and to have a vaccine. Here you have all these strategies that we are trying to use to build a vaccine for SARS-CoV-2. Okay, and except the first one, which I call the classical approaches, all the rest are based on synthetic biology. I can tell you which one is going to succeed. My personal bet would be number 6, RNA vaccines. There are several. Moderna, for example, is a very powerful company in the US that's trying to bring forward an mRNA vaccine encoding the spike protein on the surface of the virus, which is the best place probably to try to teach our immune system to detect and find and destroy the virus. But there are more than 90 projects ongoing, 5 of them already in phase 1 in clinical trials. Phase 1 means testing their safety in real human beings, not in animal models, but in humans, okay? Everybody wants a vaccine as fast as possible. Maybe we're gonna beat our record in the time that we need to produce a vaccine, but I think it will never be before next year, okay? But in any case, it could happen. And if someone comes up with a good strategy for a vaccine, then excellent. If it's this year, I would be very surprised, but I don't think that's going to happen. So the answer is, I don't know. I think we will, but I think it will take at least 1 year from now. What I can tell you is more than 90 groups are working extremely hard in the world to produce one.
Sí. Well, hello. My question was also related with COVID-19, and is that I wanted to know why it affects so much to some people and almost doesn't affect to others. Thank you.
Wow, if I knew that answer, Aina, I mean, that again is a very good question. The problem with COVID is that it produces what we call a cytokine storm. It's what more technically it's called a cytokine release syndrome, which means that your own immune system overreacts in the presence of the virus, and it overreacts especially in the lungs. Okay, so people get a respiratory distress and they need a ventilator, a mechanical ventilation, because otherwise they die. That's why so many people have died. And it affects young people, old people, people of all ages. And we don't know yet, Aina, why that happens. And I can assure you, we will know. We will know precisely why this happens. But so far, nobody has identified the common characteristic of those people that get affected. Probably it's something related to a genetic predisposition, or it's something related, I don't know. I can't assume things because the correct answer is we don't know, but we will find it.
So my question is also very related to the vaccines and COVID-19. And I was just curious, why is it so difficult to get a vaccine for COVID-19 while every year we get a vaccine for different flu viruses?
Yes, it could be, for example, because the virus mutates too quickly. But I don't think that's going to be the case in COVID, because already it is already 4 or 5 months since it was discovered. By the way, its genome was published just 15 days after being discovered, so it was a significant success again of synthetic biology and our tools. And this could be one of the reasons. But also there's something that worries me, because normal infection, there is a doubt, and this is very serious, about whether if the infection provides with good immunity after having been infected. You know, normally you get an infection, your body develops an immune response to that infection, and then you get immunity at least for 1 year, normally for 5 years or even more, against sometimes even for life, against that infection precisely. But if the virus mutates, then you lose all you had won precisely because of being immune. But in this case, in the COVID, there are significant doubts that it provides a very good immunity after having gone through the disease, and this is very bad news. If we confirm that, which again, we don't know yet, but the first results that we're getting are not very encouraging, okay? And I'm really worried. This is the #1 thing that worries me about COVID. It's not the seroprevalence, it's not the arrival of a vaccine, it's not whether we should confine or not confine. My #1 concern is precisely your question. Does it provide good immunity after infection? Because if it doesn't provide good immunity, that could be another reason why we won't find a vaccine very easily. Because if even the same virus, the virus itself doesn't provide immunity, how come will we be able to provide for that immunity? You understand? So it's tricky, but again, let's hope we will find one. I'm confident eventually we will find one. Not quickly, but we will.
Thank you.
You're welcome, Anna. Very good question.
Hello. My question is, if in the future CRISPR is used for option D, so for somatic cells diseases enhancement, could there be a case of social injustice because of the cost of it? Yeah.
Wow, what a last question. This is the most tricky and most difficult one to answer. I think there's an issue there, because you are very right. Even if it creates a little bit of social injustice, these social injustices happen only for a period of time at first. But then once the technology is well established, it gets to everyone. So I think we shouldn't be fighting that. Although I understand the question and I see your concern and I admire your concern, I think we should go ahead.
Okay, I cannot stop thinking about all the possibilities. I still remember your schematic about A, B, C, and D, and I think it's limitless. And I would like to know if you know the limits, because now I'm thinking on my field of interest, which is technology. And when you say program, I think about software. We can program the software. It's something that we cannot touch, but it's modifying, and it's giving us an outcome. And then we have in the robotics field, we have the hardware. Okay, so my question is, will we be able in the future to modify our software and our hardware as a humans?
Absolutely.
Is this included in?
Yeah, absolutely, yes. The answer is a radical yes. We will be able. You're asking if we will be able. Should we do it? That's a very different question that goes into the ethical realm, and it's very difficult to answer. But this is a problem now. I mean, if you come from the the software field, this is very similar with what is going to happen with artificial intelligence. Should we do it? Shouldn't we do it? What are the limits? Is it dangerous? Is it not? I don't know, because software, hardware, yes. But what worries me is artificial intelligence and genome editing, precisely because they transform the human race and our society in such a profound way that the changes, once they are performed, they are very difficult to take away, you know? So, but I'm very optimistic. Of course, science is a tool, as technology is a tool. It can be used for the good, it can be used for the bad. It just amplifies, you know? Is genome editing good or bad? Well, I don't know. It's an amplification of what we can do, and you can use it to amplify the good, and you can use it to amplify the bad. So it's dangerous, yes. But I'm optimistic because we will take the necessary measures to control the fallout of everything and the spread of these technologies. Because one thing is for sure, we shouldn't allow this directly without limits and without regulation. This can't go out without regulation. You know, we need to control it, and we need usually to go a little bit slower than what people want. But I'm for going slower, and I'm for doing things very carefully, but doing things after all, okay?
Pero yo digo, nosotros los profesores, ¿cómo podemos preparar a nuestros alumnos para este mundo que les viene, no sé, atravesado por la ciencia por todas partes, con los problemas éticos? ¿Qué debemos hacer los profesores para preparar a nuestros alumnos?
Gran pregunta, Corín. Yo creo que el mundo que se nos viene encima, es decir, el mundo del siglo XX, por ejemplo, era un mundo donde lo que se enseñaban eran cosas, datos, etcétera, ¿no? Es un poco, yo creo que el mundo del siglo XXI lo que hay que enseñar es precisamente enseñar a aprender cosas rápido en un entorno muy cambiante. O sea, no enseñar los datos o las cosas, sino enseñar cómo aprender esas cosas en un entorno muy cambiante y dar las herramientas a los estudiantes pues para que, sobre todo, sean hipercríticos, que sean librepensadores. El mundo del siglo XXI es un mundo de librepensadores, gente que opine por sí misma y no se deje influir por los demás, ¿no? Que estén comprometidos con la verdad y no con las fake news. El mundo está derivando hacia las fake news de manera terrible, en mi opinión, y tenemos que concienciarnos todos. Yo creo que la ciencia y los profesores, sobre todo vosotros, los científicos también, los profesores tenéis que enseñar mucho a distinguir, a que la gente sepa buscar las fuentes, sepa leer los artículos, sepa cuestionar y dudar de la palabra de los demás y solamente aceptar lo que venga de referencias en las que confíe o lo que uno mismo pueda haber entendido, etcétera. En estimular la curiosidad, y me dirás, ¿y cómo se estimula la curiosidad? Bueno, tú eres la experta, yo no, pero te digo, es lo más importante, ¿eh? Yo creo que la curiosidad es lo que mueve a la gente y lo que la hace apasionarse por las cosas. Y yo creo que si hubiera una manera de estimular, de enseñar curiosidad, que no sé si es posible, sería my number one choice.
La charla que hemos tenido ha empezado muy científica y ha acabado absolutamente humanística. Desde mi punto de vista, en los colegios lo que tenemos que hacer es justamente, yo creo que lo que ha pasado ahora mismo, que es a través de un medio tecnológico, en unas circunstancias absolutamente adversas, ha venido un experto con artículos en la mano y ha sabido enlazar a la perfección los valores éticos, humanos y a la vez la innovación científica más puntera de hoy en día. Entonces, yo dejaré terminar a Teresa, pero quería agradecerle pues esta inspiración que nos ha provocado hoy. Y estoy convencida, yo soy de letras, o sea, yo enseño lenguas, estoy convencida que hoy habrá surgido más de una vocación científica. Muchas gracias.
Gracias a vosotros, Lourdes. Y es un comentario muy, muy pertinente, porque además yo creo mucho, o sea, la ciencia es el punto de partida, pero el humanismo y el arte tienen que tener un peso muy importante para construir no solo mentes, sino personas, ¿no? Y estoy muy de acuerdo contigo que la ciencia sin humanismo y la ciencia aislada de todo, sin arte, sin sensibilidad, no es nada. Por lo tanto, creo que es la combinación de todo.